Imagine designing the perfect qPCR primers, ordering them, running your experiment, seeing beautiful amplification, but almost no fluorescence. The PCR worked. Your target DNA was amplified, yet your instrument detected almost nothing. How is that possible? Because in a TaqMan assay, primers amplify the target DNA, but the probe generates the fluorescent signal. A poorly designed probe can dramatically reduce fluorescence, increase background noise, or even make a successful PCR reaction appear to fail. The good news? Designing an effective TaqMan probe isn't complicated, once you understand the principles behind it. In this video, I'll show you exactly how I designed TaqMan probes from scratch. I'm a bioinformatician and the director of Bioinformatics. On this channel, I share practical tutorials on molecular biology and bioinformatics that you can immediately apply to your own research. Let's get started. We'll see how hydrolysis probes generate fluorescence, how to choose the best probe sequence, which design rules actually matter, and how to avoid the subtle mistakes that many researchers overlook. By the end of this video, you'll be able to design an entire TaqMan assay from primers to probe. In the previous video, our primers determined which DNA fragment would be amplified. So why can't we simply measure fluorescence using those primers alone? The answer depends on how the assay detects DNA. There are two major approaches used in real-time PCR. The first relies on intercalating dyes, such as cybergreen. These dyes bind to almost any double-stranded DNA. As more PCR product is generated, more dye binds and fluorescence increases. Simple, fast, and inexpensive. The limitation is specificity. Cybergreen doesn't distinguish between your intended amplicon, primer dimers, or non-specific PCR products. If double-stranded DNA is present, cybergreen fluoresces, whether that DNA is correct or not. TaqMan assays solve this problem differently. Instead of detecting every DNA molecule, they detect one specific sequence inside the amplicon. That's the role of the probe. Only when the probe hybridizes to its target and is cleaved during amplification does fluorescence increase. This adds an additional layer of specificity. Now your assay depends on three independent binding events: the forward primer, the reverse primer, and the probe. If any one of them fails, no fluorescent signal is produced. That's why TaqMan assays are generally more specific than dye-based assays. But that extra specificity also makes probe design critically important. So before designing one, let's first understand exactly how a TaqMan probe works. At first glance, a TaqMan probe looks like an ordinary DNA oligonucleotide, but chemically, it's much more sophisticated. In addition to its DNA sequence, the probe carries two important modifications: a fluorescent reporter, attached to the 5' end, and a quencher, attached to the 3' end. While the probe remains intact, these two molecules stay very close together. The quencher absorbs the energy emitted by the reporter, preventing fluorescent light from reaching the detector. This phenomenon is called fluorescence quenching. So even though the reporter is continuously excited by the instrument's light source, almost no fluorescence is detected. Now let's follow one PCR cycle. The DNA is first denatured, primers anneal to their target sequences. The probe also hybridizes within the amplicon. Everything is now in position. DNA polymerase begins extending from the primer as it synthesizes the new DNA strand. It eventually reaches the bound probe. As it synthesizes the complementary strand, it naturally encounters the probe positioned ahead of it. This allows the enzyme's 5' to 3' exonuclease activity to cleave the probe during extension and release the fluorescent reporter. When polymerase reaches the bound probe, its 5' to 3' exonuclease activity cleaves the probe rather than simply displacing it. As the probe is cleaved, the reporter becomes physically separated from the quencher. Once that distance increases, quenching can no longer occur. The reporter is finally free to emit fluorescence. The instrument detects that light. With every PCR cycle, more probe molecules are cleaved, more reporter molecules are released, and fluorescence continues to increase. This is what makes TaqMan assays so specific. Fluorescence is generated only when the correct target sequence is recognized, bound by the probe, and cleaved during amplification. Understanding this mechanism makes nearly every probe design rule much easier to understand. The first design decision is surprisingly simple. Where should the probe be located? The answer is straightforward: inside the amplicon, between the two primers. In most assays, the probe is also positioned on the strand that will be encountered by polymerase extending from the forward primer, never outside the amplified region and never overlapping either primer. Think of your amplicon as a bridge. The primers define its two ends. The probe belongs somewhere in the middle. This arrangement allows DNA polymerase to encounter the probe naturally as it extends the new strand. If the probe overlaps either primer, the two oligonucleotides compete for the same target sequence. That competition reduces binding efficiency and can significantly decrease fluorescence. Likewise, placing the probe extremely close to either primer isn't ideal. Leaving a small gap improves hybridization and minimizes steric interference during extension. Fortunately, primer 3+ usually suggests suitable probe positions automatically, but software isn't perfect. Always inspect the final design yourself. Unlike primers, probes don't initiate DNA synthesis. Their job is simply to recognize and remain bound to the target sequence until polymerase reaches them. For that reason, they're usually slightly longer than primers. A good starting range is approximately 20 to 30 nucleotides. Most successful TaqMan assays fall comfortably within this interval. If a probe is too short, its melting temperature may be too low, making hybridization unstable. If it's too long, specificity may decrease, and the likelihood of unwanted secondary structures increases. Longer isn't necessarily better. The goal is stable, specific hybridization under your PCR conditions. As we'll see next, probe length and melting temperature should always be considered together. If I could evaluate only one parameter during probe design, it would be the melting temperature. Why? Because the probe must remain hybridized while DNA polymerase is actively synthesizing the new strand. In most qPCR assays, primers have melting temperatures close to 60 degrees Celsius. Your probe should typically melt about 8 to 10 degrees higher, usually around 68 to 70 degrees Celsius. That difference is intentional. Once DNA polymerase begins extension, the primers have already completed their job. The probe's job is just beginning. It must stay bound long enough to be recognized and cleaved by the polymerase. If the probe dissociates too early, polymerase simply passes through the target region without cleaving it, resulting in much weaker fluorescence. That's one of the most frustrating problems in TaqMan assay design. The amplification may be perfectly successful, yet the fluorescence signal is unexpectedly low. Maintaining a higher probe melting temperature greatly reduces that risk. Now that we've selected an approximate position, let's optimize the probe sequence itself. Probe design is generally less forgiving than primer design. A primer with minor imperfections may still amplify efficiently. A poorly designed probe, however, often produces weak fluorescence, poor reproducibility, or reduced assay sensitivity. Let's examine the sequence one rule at a time. Rule 1. Maintain balanced GC content like primers. The probe should generally have a GC content between 40 and 60%. Too little GC, and hybridization becomes unstable. Too much GC in the probe may bind too tightly or form stable secondary structures. The objective isn't maximum binding strength. It's stable and specific hybridization under PCR conditions. Rule 2. Avoid long homopolymer runs next. Look for repeated nucleotides. 5A5T, 5G, 5C. Long homopolymer stretches may appear harmless, but they often create unexpected problems. They can promote secondary structures, reduce oligonucleotide synthesis quality, and sometimes increase background fluorescence. Whenever possible, avoid runs longer than 4 identical nucleotides. If 2 probe candidates perform similarly, choose the sequence with the more balanced nucleotide composition. Rule 3. Avoid G at the 5' end. This is probably the best known rule in TaqMan probe design. Whenever possible, avoid placing guanine at the 5' end of the probe. Why? Because many reporter dyes, particularly FAM, are partially quenched by an adjacent guanine residue. As a result, even after probe cleavage, the fluorescent signal may be lower than expected. Your assay may still work perfectly, but its sensitivity can be reduced by this single nucleotide. Fortunately, it's also one of the easiest design mistakes to avoid. Rule 4. Avoid excessive GC clusters at the ends. 1 or 2 GC bases near either end are perfectly acceptable. However, large GC-rich clusters at the probe ends are usually undesirable. They can encourage unwanted secondary structures and alter hybridization kinetics. Unlike primers, a probe doesn't need a GC clamp. Its role is target recognition, not extension. For probes, balanced sequence composition is far more important than maximizing end stability. Now let's design the probe. Open Primer 3+, the same software we use for primer design. Paste exactly the same template sequence. This time, enable internal oligo. That's Primer 3+'s name for the hydrolysis probe. Several additional settings will appear. Most of the default values are already reasonable, but I usually adjust a few of them. First, set the probe length. A minimum of about 20 nucleotides, an optimum around 25, and a maximum close to 30. Works well for most assays. Next, adjust the probe melting temperature. Since our primers are close to 60 degrees Celsius, set the optimum probe TM to approximately 69 or 7 degrees Celsius. Finally, allow Primer 3+ to generate multiple probe candidates. Don't limit yourself to a single suggestion. Having several candidates makes it much easier to choose the best sequence. Click Pick Primers. Within a few seconds, Primer 3+ will generate primer pairs together with their corresponding probe candidates. Now the real evaluation begins. When comparing probe candidates, I don't start with the software score. I start with the sequence itself. First, does the probe begin with a G? If it does, I usually move on to the next candidate. Second, is the melting temperature roughly 8 to 10 degrees higher than the primers? Third, is the probe positioned appropriately within the amplicon? Fourth, does the sequence have balanced GC content without long homopolymer runs or any obvious sequence problems? Only after those questions are answered, do I compare the software ranking. Primer 3+ evaluates candidates mathematically. You still need to evaluate them biologically. A probe that looks perfect computationally may perform poorly if it targets a highly variable region of the genome. That's why I always ask one additional question before making a final decision. Is this target region genetically stable? Even a single nucleotide difference within the probe binding site can noticeably reduce hybridization efficiency. In some cases, one mismatch is enough to delay fluorescence by several PCR cycles. This becomes especially important when working with clinical samples, genetically diverse populations, or rapidly evolving pathogens. Whenever possible, choose a highly conserved target region for human assays. This is easy to verify. Simply identify the genomic region where your probe binds. Then check databases such as NCBI-DB-SNP or Genome Data Viewer for known sequence variants. If a common SNP falls within the probe binding site, particularly near the center of the probe, it's usually better to choose another candidate. If you're designing assays for viruses or other highly variable organisms, don't rely on a single reference sequence. Instead, Align multiple sequences using tools such as Clustal Omega or MAFT, and design your probe within the most conserved region available. Spending just a few minutes checking sequence variation before ordering can save hours of troubleshooting later. Another common question is, should probes be analyzed with oligoanalyzer, just like primers? Absolutely. Although probes are not extended by DNA polymerase, they still need to hybridize efficiently to their target. Hairpins, self-dimers, and other stable secondary structures reduce the number of probe molecules available for target binding. The result can be lower fluorescence, greater CT variability, and reduced assay sensitivity. Fortunately, the evaluation process is exactly the same as it is for primers. Paste the probe sequence into oligoanalyzer. Check for stable hairpins. Check for strong self-dimers. Cross-dimers are usually much less important for a single probe. But stable internal structures should always be avoided. Pay particular attention to structures with highly negative delta G values. If two probe candidates are otherwise similar, choose the one with the simpler secondary structure. A probe should spend its time binding your target, not itself. At this point, our probe sequence is complete, but before ordering it, there's one more important decision. Which fluorescent chemistry should you choose? Unlike primers, a TaqMan probe isn't just a DNA sequence. It also contains chemical modifications that determine how fluorescence is generated and detected. Let's start with the reporter dye. The reporter is attached to the 5' end of the probe. This is the molecule that emits fluorescence after the probe is cleaved. For most single-target qPCR assays, the standard choice is FAM. It's bright, highly sensitive, supported by virtually every real-time PCR instrument, and compatible with most commercial master mixes. If this is your first TaqMan assay, FAM is usually the safest choice. But sometimes, one fluorescent color isn't enough. If you want to detect 2 or more targets in the same reaction, each probe must carry a different reporter dye. Otherwise, your instrument can't distinguish one target from another. Common alternatives include Hex, Vic, Psi-5, Texas Red, and ROX. Each emits light at a different wavelength, allowing multiple targets to be detected simultaneously. Before choosing a fluorophore, always verify that your qPCR instrument supports the corresponding detection channel. A perfectly designed probe is useless if your instrument can't detect its fluorescence. The opposite end of the probe carries the quencher. Its job is simple: suppress fluorescence until the probe is cleaved. Modern probes typically use dark quenchers. Unlike older fluorescent quenchers, they absorb the reporter's energy without producing their own fluorescent signal. The result is lower background and a better signal-to-noise ratio. Common examples include BHQ1, BHQ2, Iowa Black, and NFQ for most FAM-labeled probes. BHQ1 remains an excellent all-purpose choice. What about MGB? You may also encounter probes labeled MGB, which stands for minor groove binder. This small molecule increases probe binding stability, allowing shorter probes to maintain high melting temperatures. That's especially useful when detecting SNPs, point mutations, or closely related target sequences. Shorter probes generally discriminate single base mismatches more effectively for routine gene expression assays. A conventional probe is usually sufficient, but for SNP genotyping or mutation detection, an MGB probe is often the better option. Before clicking order, take one final look at your assay. Ask yourself, is the probe located completely between the primers? Does it avoid overlapping either primer? Is its length approximately 20-30 nucleotides? Is the melting temperature about 8-10 degrees Celsius higher than the primers? Is the GC content between 40 and 60%? Does the probe avoid a 5' guanine? Are long homopolymer runs absent? Are hairpins and self-dimers minimal? Is the target region highly conserved? Have common SNPs been excluded from the probe binding site? Have you selected an appropriate reporter dye? Is the quencher compatible with that reporter? And is the fluorophore supported by your qPCR instrument? If every answer is yes, your probe is ready for synthesis. If you found these 2 videos helpful, consider subscribing. In the next tutorials, we'll move beyond assay design and focus on optimization and troubleshooting. You'll learn how to interpret amplification curves, analyze melt curves, recognize primer-dimers, calculate PCR efficiency, and troubleshoot the most common qPCR problems encountered in the laboratory. Thanks for watching, and if you're working on a research project and need help with qPCR assay design or other bioinformatics analyses, feel free to visit bioinformatics.ca to learn more about our services. I'll see you in the next video.